My name is Dr. Robin Matham, and this short video will take you through the current state of COVID diagnostics. So, this diagram is from Analytical and Bioanalytical Chemistry, and it provides a good overview of the current state of diagnostics, and we're going to walk through this step by step. At the beginning, we start off with sample collection. This is done by nasopharyngeal swab most commonly, the swab which is put into the back of your throat and up your nose to take a sample of cells which you expect to potentially be infected by virus. The swab is placed into a sample storage medium, and for sample storage medium you have two effective choices at the moment. You either try and keep the sample relatively intact, so if it's put through the postage system or whatever, the virus is kept in a reasonable state to be analysed, or you have a medium which is designed to lyse the virus to release components for analysis, and potentially, if you're going to do that immediately, you also want something that will stabilise the virus so that the parts of it don't get degraded so you can still detect it. Now, the most popular way at the moment of testing is antigen-based assays, so we're going to have a look at that first. And what these do is they detect bits of the virus, proteins that are used either on the viral surface or inside the virus as part of the packaging of the virus. To do this, you disrupt the structure of the virus so these proteins are released. The antigen is then captured on an antibody when you put your sample into the strip. It flows down. If it binds to the target, then you will have the presence of the antigen detected. You have a second antibody there with a label attached. Often this is a nano gold particle that allows you to see what's going on, and this will then light up the test strip if you're positive for virus. There is a second control strip there to show that the assay's worked, and what this does is detect that you've actually put a sample in and it's flowed down the strip correctly, and you've picked up the presence of a sample being put in the test correctly. These test strips work very much like a pregnancy test. It's a yes or no. Do you see the strip highlighted or don't you? If the control strip doesn't work, you need to do the test again. Now, there's another way we can sample the virus, and that's not to detect virus proteins, but to detect the RNA, the genetic material of the virus that is present inside that. And we can do that in two ways. We either take it straight from the transport medium in an extraction-free protocol to a step of reverse transcription. This is turning the viral RNA into a complementary cDNA copy, which we need for molecular detection techniques. The other way we do this is we actually lyse up the virus and remove all of the junk, the viral proteins, the cell proteins, all the rest of the stuff that's present in your swab sample, and then take it through a purification step to end up with purified RNA to put into this same reverse transcriptase reaction. This has the advantage that you get rid of things that may interfere with this reverse transcription step. The extraction-free protocols are faster, but potentially suffer from inhibitors that may be present in the sample which could stop the reaction from working. So these are quicker and faster, but potentially less reliable than the lysis and RNA extraction steps, which are generally done for most molecular testing. Okay, so let's then move on and see what we can do with molecular testing. Well, one of the first things we can do with molecular testing is a similar sort of lateral flow assay. And once we've reverse transcribed our RNA into DNA, we can then amplify it, and there's a detection test which works in a similar way to the lateral flow test we've just seen. We've got an amplified part of the viral genome, amplified by a process called LAMP, loop mediated amplification. This uses a clever piece of enzymology with a CRISPR-Cas system which recognises the amplified strand and uses it to bind a target to, which then hydrolyzes a single strand DNA probe and releases a fluorescence that allows you to show that you've actually got target there. So this is again a lateral flow test and a strip where you're looking to see, does the strip light up or not? That's another yes no answer, but this is a molecular based test using the viral RNA rather than viral antigens, the proteins present in the virus. The last way is the most common way and the gold standard way of molecular detection, which is via PCR amplification. So we take our amplified DNA, we use the polymerase chain reaction to amplify this up in a quantitative matter, qPCR, which amplifies it in the presence of a fluorophore, which detects the amount of viral RNA which is present and been turned into DNA and is amplified at each step. And what this allows us to do is to get this sort of graphical readout of fluorescence, which tells us how much we've got amplified over a number of copies, a number of cycles that we've done. The longer it takes us to amplify the target, the less we started with. So theoretically, this has a very sensitive detection down to potentially a single molecule of viral RNA in the sample, up to millions of copies. And the big thing about this qPCR amplification is it gives us a quantitative answer, how much virus has somebody got in their system. Downstream of PCR amplification, we can also amplify the RNA to allow us to sequence it, and this has become very important these days because we wish to be able to sequence viral RNA positive samples so that we can detect variations in the sequence that allows us to say, is this a variant, is it the alpha variant, is it the delta variant. Sequencing can be done by a couple of different methods, sequencing by synthesis or nanopore sequencing. Both of these rely on first preparation of a DNA amplified library of the original RNA to allow sequencing. And we won't go into any more detail about this sort of sequencing at this stage. The last molecular technique, or the other molecular technique, is loop-mediated amplification. Now, we talked about this in terms of the ability to do this lateral flow type test, but the other thing we can do is use this amplification procedure to provide a colorimetric readout. And what happens is, as you amplify up the viral RNA copied into DNA, the amplification process can be used to produce a colour change in a reaction tube. If it's negative, it stays red. If it's positive, the reaction will turn yellow due to the pH change produced by the amplification of the DNA. This is a molecular technique, but it's taken to be a yes-no molecular technique, as opposed to the qPCR amplification, which is considered to be a quantitative technique. So this will tell you how much viral RNA you started with. This just says, did you have it or did you not have it? Now, this becomes a little bit more problematic when you're thinking about samples with low amounts of virus in it. Can you detect them? Can you differentiate them with people who don't have any virus or have lots of virus? So actually what we want to be able to do is go from a qualitative to a quantitative system, and there are ways of running LAMP reactions which become more like the gold standard qPCR and give us a more quantitative readout of the amount of viral RNA in a sample. So that provides us an overview of the whole process, and hopefully you can now differentiate between our antigen-based techniques, molecular lateral flow tests, molecular techniques and the sequencing, which is important for detection of variants, and our LAMP test, which may be able to become more quantitative rather than qualitative with the right sort of technology.